Well, good morning, everybody. My name is Nilo Azad, and it is my pleasure to open this clinical trials plenary session today, where today we're really going to highlight the work of 4 of our colleagues and their teams in work that they've done that is really going to advance the possibility of immunotherapy and PD-1 therapy for our patients. I think it's very appropriate that this session is dedicated to Dr. Waun Hong, who is a pioneer in the field of cancer research who we unfortunately lost just a few months ago. Dr. Waun Hong was a classic example of the American dream. He came to the United States in 1970 after serving as a flight surgeon in Korea, and he came as an immigrant who spoke little English and with little resources, and over the course of 50 years developed a program and a legacy of care for patients with head and neck and lung cancer that is really unparalleled. After training in New York, he eventually landed at MD Anderson. And at that institution, his work included pivotal work that changed the way that we take care of patients with laryngeal cancer, showing that chemoradiation would be as effective as surgery, trials in cancer prevention, and even trials in molecularly targeted therapy, the battle study in lung cancer where he served as the PI. I think he would have been so pleased today to see these presentations, including work in HPV-positive cancer, which was very important to his legacy as well. And with that, I'm going to introduce Ryan Sullivan, who's going to give us our first data presentation, the efficacy of entinostat and pembrolizumab in patients with melanoma previously treated with anti-PD-1 therapy. Thank you, Dr. Sullivan.
Thank you very much. It's a pleasure to be here and to be part of this session. Dr. Hong's name. I will ultimately get to my files. Okay, excellent. So I'm here today on behalf of my co-investigators and study sponsors to report the efficacy and safety of entinostat and pembrolizumab in patients with melanoma previously treated with anti-PD-1 therapy. These are my relevant disclosures. So, in part, maybe it's because I was next to the College Football Hall of Fame, but when looking at the advances in melanoma therapy over the past decade, I sort of want to spike the football because we've made such great progress in a short period of time. In orange are BRAF-targeted therapies, both single agent and 3 combinations of MEK and BRAF inhibitor therapies. In blue or aqua are the immunotherapies, including the immune checkpoint inhibitors, ipilimumab, nivolumab, and pembrolizumab, as well as an oncolytic herpes virus. And then the combination of ipilimumab and nivolumab. All of these have impacted the lives of our patients and are filled our clinics with follow-up visits with patients who remain disease-free for many years. In fact, over time, you can even chart out how many patients are, we dare say, cured to patients in general, but we can say cured in places like this, at least as far as the follow-up we have. With high-dose interleukin-2, that rate was probably around 10%. With ipilimumab, the long-term survival is about 20%, and as long-term survival is being reported with the anti-PD-1 antibodies, either as single agent or in combination, we're thinking that that rate of cure will probably be somewhere in the 30 to 40% range. And yet, despite all that progress, and this is data from the CheckMate 067 study, most patients develop resistance, and this is the overall survival data. Most patients will still die of metastatic melanoma, despite all those advances. And so I think it's always important to define what the unmet need is, and here it's actually quite clear. Most patients are not receiving ultimate benefit, and as a result, we need a better therapeutic approach. Now, in addressing the unmet need, there are a few things that we can do that can help us. The first we can do is try and improve our understanding of the mechanisms of therapeutic resistance. And so I'll just spend 30 seconds or so describing. So there's lots of different pictures of doing this. I have to thank David Liu, who's now at Dana-Farber, who beautifully drew this and probably spent a long time doing it. But just really showing the cycle of the immune and tumor interaction. And at a lot of these different points, there can arise resistance mechanisms. It can be just the fact that those tumors aren't particularly well recognized, that they have low neoantigens or tumor mutational burden. There could be insufficient priming. There can be inadequate T-cell recruitment or infiltration. There can be loss of tumor antigen presentation machinery or loss of interferon signaling. Alternative immune checkpoint expression may also play a role. And importantly, and perhaps, and certainly it's going to be a topic that we're going to come back to in this talk, intratumoral cellular components may regulate the immune response and in fact downregulate it. And those include myeloid-derived suppressor cells and T regulatory cells. And so with a better understanding of the mechanisms of resistance, we may be able to develop more effective therapies. And though the frontline setting is a critical place for us to be in terms of clinical trials, I think certainly the most relevant and most unmet need now is what do we do with patients who have received anti-PD-1 antibody therapy and need something else. The rationale for entinostat is described here. Essentially, entinostat, which is a class 1 selective histone deacetylase inhibitor, has effects on myeloid-derived suppressor cells. This is showing entinostat in a model where MDSCs are mixed co-culture with T cells, and you're just looking at the proliferation of T cells. And when you have higher ratio, more MDSCs to T cells, you end up getting more suppression of T cell function. Entinostat relieves that at each of the ratios that are presented here. In addition, entinostat also down-modulates RNA expression of a number of key genes that are important for regulatory suppression, including INOS, ARG1, and COX2, and in a xenograph model, and you see really quite robust synergy between entinostat and an anti-PD-1 antibody. Interestingly, in patients treated with entinostat on a phase 1 study, there was a monitoring of MDSCs that was part of the correlative work, showing a pretty consistent down-modulation of circulating peripheral MDSCs after a single dose of therapy 14 days after the dose. And so with all of that, the combination of entinostat plus pembrolizumab was studied in the ENCORE 601 trial. Dose and safety confirmation occurred, and then there was an opening of 4 phase 2 expansion cohorts, 2 in non-small cell lung cancer, 1 in mismatch repair proficient colorectal cancer, and the cohort that I'm going to be discussing today, which is the melanoma patients who had progressed on or after anti-PD-1 antibody therapy. The inclusion criteria are here, or at least the key ones: a recurrent or metastatic melanoma measurable by RECIST 1.1, prior progression on and after anti-PD-1 or PD-L1 therapy, prior BRAF treatment if patients had a BRAF V600 mutation, an ECOG performance status of less than 2, and a willingness to participate in baseline and on-treatment biopsy and blood samples. The primary endpoint was objective response rate, which was defined by IR RECIST, and a number of secondary endpoints, including clinical benefit rate defined by stable disease greater than or equal to 6 months plus response, progression-free survival, overall survival, and safety and tolerability. And all 53 patients were enrolled, and the last patient enrolled was in April of 2018. The baseline demographic data is here. It's a little busy, but basically this is a pretty standard metastatic melanoma population. More males than females, median age in the early 60s. Performance status was mostly 0, but a significant minority were ECOG 1. About half of patients were positive for PD-L1. The majority had visceral metastases, and a significant minority had elevation of LDH. With regards to prior therapy, 70% had received both a PD-1 inhibitor and ipilimumab, either in combination, which is about 35% of patients, or in sequence. And again, all patients who were BRAF mutated were required to have BRAF MEK therapy, and that rate of patients on this study was 23%. Key issues about the prior PD-1 history, there was a response rate that was 13%, much lower than you would imagine for all comers getting frontline PD-1 inhibitor therapy in melanoma, and truly patients that you would think would need something else. And the median duration between PD-1 therapy, your last dose of PD-1 therapy, and first dose of entinostat was 2.6 months. Here is the change in tumor volume. The majority of patients had some tumor regression, but the overall response rate was 19%, with 10 confirmed responders of the 53 patients, with 1 complete responder and 9 partial responders. The duration of response was 13 months, at least at median, and 4 responders have an ongoing response. Additionally, 9 patients had stable disease that lasted for greater than 6 months, and so the clinical benefit rate, adding the responders in those patients, was 36%. Here's a spider plot again demonstrating that those responders who are in green tend to have prolonged benefit from therapy. Shown a different way in the swimmer lane plot. Importantly, at the 1-year mark, 10 patients remained either on therapy or in response. This is a patient who actually got very short course of therapy, developed significant hepatitis, had a response, and that response has been maintained off of therapy for well over a year. In addition, 4 patients remain on active therapy, and a few other patients have come off of therapy and are being followed. This is the prior therapy that those who responded received. On this side of the line is the prior therapy, and this side of the line is the time on study. There are certainly some patients who received their PD-1 inhibitor long before being enrolled onto this study and ultimately responded, but there are also a number of these patients who received PD-1 inhibitor right before going on to study, and obviously were defined as progressors. And so clearly it seemed to be helping both those who remotely were treated with PD-1 inhibitors and those who were recently treated with PD-1 inhibitors. Here is the safety treatment data. So it's presented as all toxicities that happened greater than or equal to 15% of the time, as well as all grade 3 or 4 toxicity that happened in more than 1 patient. The predominant toxicities that we saw at levels of greater than 15% included nausea, fatigue, diarrhea, myelosuppression with either a reduced neutrophil count or platelet count or white blood cell count. And 6 patients discontinued due to related AEs. And importantly, there were only 5 grade 3 or 4 immune-related AEs. I mentioned the patient had autoimmune hepatitis, but there was a case of pneumonitis, a case of colitis, and 2 cases of significant dermatitis. This figure shows some of the correlative data. So in 49 of the 53 patients, there was peripheral blood mononuclear cells for analysis available, and this shows that there wasn't a significant change in T-cell populations across responders in blue and non-responders in red at cycle 1, day 1, cycle 2, day 1, cycle 2, day 15. However, with the peripheral blood monocytic myeloid-derived suppressor cells, defined here is, we see a significant reduction early and ongoing over time in the responders. There was a slight reduction as well in the non-responders. In addition, this is the responders, and every patient but one had a significant reduction over time, and it continued even through cycle 2, day 1. So at cycle 2, day 1, it's here, and then past cycle 2, day 1 to cycle 2, day 15, it was even more prominent. There was a pre- and on-treatment biopsies available for RNA sequencing in 8 patients. We compared 4 responders and 4 non-responders. In comparison of the responders and non-responders, there were some things that you might expect enriched in responders, like an inflammatory response gene signature. But then there are other things that are interesting and potentially hypothesis generating, but hard to wrap your head around a little bit, like there was more hypoxia, more EMT, TNF-alpha signaling. In the gene sets that were enriched in the non-responders, these include E2F targets, G2M checkpoint, oxidative phosphorylation of fatty acid metabolism. Now, when we looked at the responders, post-treatment versus pre-treatment, we actually saw something very similar. In the patients who responded, there was a further enrichment of EMT and then further enrichment of immune gene signatures like allograft rejection, interferon gamma, IL-6, JAK, and STAT3 signaling, and UV response. And those that were down-modulated were very similar to those that were enriched in the non-responders, E2F targets, G2M checkpoint, MYC targets, and OXPHOS. When looking at individual genes from the RNA sequencing and comparing in the patients who responded post-treatment versus pre-treatment, a number of genes were highlighted as significantly upregulated, and these include several immune-related genes like CCL21, CXCL9, DPP4, a number of immunoglobulin markers, and IL-15RA. Interestingly, there was only one gene that was significantly down-modulated in the responders over time, and that's CXCR1. CXCR1 is actually a gene that's been associated with melanoma progression. And so it's interesting in that these folks who are responding, and I guess not entirely surprising that a gene associated with melanoma progression would actually go down in responders, but it's at least nice to see that and is consistent with the theme. This is nanostring analysis that was performed on 7 patients, 3 responders and 4 non-responders, and I'm just showing the genes, the immune signatures that generally, but then also the ones that significantly changed from post-treatment to pre-treatment. These include things like antigen presentation machinery, lymphoid signatures, interferon gamma signatures, CD8 positive T cells and cytotoxic T cells. Interestingly, though these aren't significantly decreased, myeloid inflammation and DC were hits on this. In the volcano plot, interestingly, one of the top genes to come up is beta-2 microglobulin. One of the other potential mechanisms of how entinostat could work is that it could increase antigen expression in tumors that may have down-modulated antigen expression. And so to see a gene like beta-2M coming up over time is encouraging with an alternative mechanism that goes beyond just affecting myeloid or T regulatory cells. So in conclusion, patients with progressing melanoma on prior PD-1 blockade, including prior PD-1 and anti-CTLA-4 blockade, clearly a group with limited treatment options. Entinostat plus pembrolizumab demonstrated significant anti-tumor activity. The combination of entinostat plus pembrolizumab was clearly safe and tolerable, and in fact, the dominant toxicity was additive toxicity from entinostat, and no apparent increase in IRAEs with the combination. The preliminary biomarker analysis, and it's very preliminary on a small number of patients, demonstrates findings consistent with the mechanisms of action of entinostat, including reduction in circulating myeloid-derived suppressor cells, as well as tumor-specific increases in the pre and on treatment and enriched in the responders versus non-responders inflammatory pathways. As always, in doing clinical trials, there's lots of people to acknowledge, most importantly the patients and their families, my co-investigators and their study staffs, Rancho Biosciences and Dan Rosell, who coordinated the RNA sequencing efforts at NanoString, Amy Sullivan, Sarah Church, who helped do the NanoString analysis, the Wistar Institute, Fang Wang, who did the circulating PBMC analysis in Dmitry Gabrilovich's lab, and then our study sponsors, Syndex Pharmaceuticals and Merck. With that, I also thank you for your attention. I'm happy to answer any questions.
So Dr. Sullivan, I'm going to invite you to join me on stage, and then we're going to introduce Dr. Steve Balin, who is an internationally renowned epigeneticist for his cogent discussion on this data as well. And then it would be wonderful to be able to take some questions.
Well, thank you, Nilo. It's an honor to be asked to do this, and I will try to, in a pithy way, summarize some things about this trial. My discussion should be right here as I see the screen. I don't know about internationally renowned, but I am internationally continually surprised by what's happening in this field since we entered it. And it is pleasing to see this trial indeed. Let's see. Okay, Nilo, you have a hint what I'm doing? All right. So, since we entered the field some 8 years ago with collision of epigenetic therapy with PD-1 and other labs at the time, it's been about, I guess, 8 years through some chance collisions, and things are indeed changing. So, a promissory note has been on the list for some time that epigenetic therapy could be one of many ways, and certainly all the ways are not listed here, to improve the efficacy of immune checkpoint therapy, as you heard. Initially, the promise was being sought in a more frontline setting in advanced patients. This question of reversing resistance has galvanized the field, I think, and may be the most important use of the epigenetic therapy for some of the things that we would like to see. Now, this is an important trial, and the positive findings of this trial are pivotal at a pivotal time in the field because we need to get forward momentum over a promissory note. And it's very impressive in this setting to see reversing of resistance that involves RECIST criteria responses. Like, this is fantastic. The overall medical benefit that was seen here in this trial is impressive to me, and I think very positive for seeing the overall potential role for epigenetic therapy in the setting of immunotherapy. Right now, I want to remind the audience, to my knowledge, there is no approved epigenetic therapy agent for solid tumors. They are FDA approved for hematologic malignancies, including HDAC inhibitors. So this is a very important issue I think we need to consider. So there are questions that have been raised and well discussed by the presenter, and I'd like to introduce them and then expand on a few of them. So why are only 4 of 10 responders still ongoing? And is this some sort of effect that would suggest this isn't going to be durable? I'm going to make the cases that this may not be that much of a downer in this setting, and I'll make some points about that. There were a very wide range of times between the patients receiving their initial immune checkpoint therapy and the institution of the combined therapy with the atinostat. Is that a mystery? Is that a bad thing that it was that variable? It's been alluded to, and I'll make the point, that the initial effects of epigenetic therapy as events in antigen stimulation of cells, as I understand them, are occurring. You can get a very initial response that can be very long-lasting. And I think this is a very important point, and I'll come back to it a little bit. Where are the prime cellular components for the beneficial action of the atinostat in this regard? This is really important, and I'm going to comment on it. I think it's quite interesting and important preliminary data, cellular and molecular in this regard, and I'm going to come back to some aspects of that. So this is an overview about epigenetic therapy in cancer and immune checkpoint therapy from a review by myself and Peter Jones, and I want to make a couple of points here. I think we have a synthesis of effects we want to establish with epigenetic therapy that may really be the most important for seeing efficacy in this arena. First, let's look at the tumor cell compartment. For some time, it's been known that DNA demethylating agents and HDAC inhibitors can reverse the silencing of tumor suppressor genes, which is epigenetically mediated, and other key genes. And if you can add that to what's just been shown for the effect of zoltinostat or any other agents. No tumor is going to like it. In point of fact, the drugs that we have to use right now, they're really interesting drugs like azacitidine and the DNA demethylating, and to some extent HDAC inhibitors, are very proliferation dependent. And you can imagine that in many solid tumors, you have a very small percentage of cycling cells at any given time. And I think that's been limiting and makes this compartment pretty difficult. If we can add it with better drugs and come into it, it's going to join this immune compartment because these are joined issues. That's why I call that the triad. The tumor effects, the immune cells and the immune effects, and the joining between these that many genes could mediate. We need to do better here. It's now been observed that we're seemingly doing better here in animal models, syngeneic, and even in the patients in seeing the immune effects. Maybe because we're getting para effects. We get a few cells that are exposed, but they can emit substances that recruit the rest of the tumor cells into responses that are very important in this setting. And so that's why. And I'm going to comment on the fact that in our hands, one of the things that intrigued us early on in Dan DiCarvalho's lab when he left Peter Jones's group is this signaling that comes about through things like endogenous retroviruses and repeat sequences that make the tumor cells, at least, seem like they've seen a virus and are responding in a protective way to that virus to generate interferon responses. We're seeing more and more that some of this also goes on in the immune subset of cells. So this is all going to be a very important, I think, joint series of events. Now, I'm a little surprised at one thing, but perhaps shouldn't be. In our hands, all the work that we've done, including now in syngeneic models, things have worked better when we add a DNA methyltransferase inhibitor to the HDAC inhibitor. But the HDAC inhibitors are a really important part of the complex. And what's emerging, I think, is that they alone, as you've heard now, can exert key effects. This is a paper just about a little over a year ago from Michael Topper and Michelle Vaz in our group, showing you a synthesis of some of the things we've seen combining these two agents in two syngeneic models of non-small cell lung cancer. And what we've seen is that they work together to augment the signaling for this interferon signaling cascade I told you about. And as you heard, importantly, downstream from this interferon signaling are many things and steps that are important. One being antigen presentation and upregulation of ability of MHC complexes to carry antigens. This is really critical. In this model, in the non-small cell, what we saw are key cytokines. In this case, what seemed really important was the upregulation of CCL5. And that in this model was associated with the fact that many of these epigenetic therapies really look good for bringing activated T cells into the tumor, carrying interferon gamma and key effector states of these cells. That's very important. What we also saw, and this is about my comment about the states of the immune cells we need to see, is that John Ware's transcriptomic profiling for the TILs in this study, post-treatment in the tumors, profoundly reversed exhaustion to a non-exhaustion transcriptomic state. So if the genes in exhaustion were down, they went up. If they and reverse, and this was very robustly statistically significant. And there are key hints that this may be a key part of the players for HDAC inhibitors, DNA methyltransferase singly or together. And importantly, what we observed here was, and it seemed in this case the DMTI may sensitize the HDAC inhibitor to do this, was the leading downregulation pathway was MYC and its targets. And you saw that listed here. This is critical. I think we often forget it, but in melanoma, the Wnt pathway and MYC, and the MYC itself directly repressed in this study the ability to upregulate this interferon signaling pathway. And so you get a twofer with these drugs where you're downregulating proliferation of the tumors and MYC, which is inverse and to allowing signaling that you might like to get for immune attraction and the T cell responses. And it's important that in a paper just a month after this one was published from our group, Gerard Evans, in one of the same non-small cell mine, manipulated MYC genetically and got very similar effects. And so I also want to remind you, generating inflammation in the tumor tissue is really critical. and told about that. And the viral mimicry turns out to be just a significant part of an inflammasome alone. And on Tuesday, Fayrouz Rasool's lab is going to present this amplification in this whole inflammasome signal, and I urge you to go see that. But when you get this in the tumor, it pretty quickly starts to signal, as we're talking about, to the immune cells. And the immune interactions substantially are an epigenetically related series of events. Very exciting as we're learning about it. So you not only activate CD8 T cells on this basis, but proteins that they carry, such as CX3CR1, are controlled by DNA methylation. The drugs affect all of this. That receptor brings cells to the vascular epithelium, interacting with fractalins that help them get there and invade, and then all of this signaling starts to occur again between the CD8 cells that got there and the periphery, and it's our real epigenetic cascade. One final point. This is a trial from Julie Bremer and Kristen Marone that we started some years ago that's still ongoing. I think it's the longest one now, combining tinostat and Vidasa, a DMTI from Celgene. And I want to make a couple of final points to it that address, I think, some of the issues we saw here. So on this side, we have an arm for nivolumab alone, and this side combines the epigenetic therapy with nivolumab, and survival is listed in weeks on the graph right here. Both of these drugs, nivolumab and the Vidasa and the tinostat, cause about the same 30% of long-term survival for these patients, going out some 4 to 5 years. So they both can do it. What's interesting is there are more PRs and CRs in the nivolumab, but most of them don't go on that long. And I think this addresses a key issue we just heard about. In this trial, we have very few RECIST criteria responses, and yet we've got these 7 individuals who lived past 2 years. 5 of 7 are still alive, as shown by the pink bars, 1 now out almost 4 years, and the others. And so long-term survival may be the metric that really is going to read this out, I think, in the most robust way, rather than just RECIST criteria responses. A final note, in the combination arm, these patients, as we sequence their tumors, have a low tumor mutation burden and almost no PD-L1 relative to these people. So it raises the issue, are there different reasons for long-term survival with immune checkpoint monotherapy versus what you might have with epigenetic therapy? And could we really spot these in biopsies and early on know which patients are going to go which ways and personalize all of this? And so I conclude this is a very important trial, stresses the metrics we need to keep exploring, the best clinical outcomes to expect, overall survival. Could it be somewhat independent of RECIST criteria responses? What are the best drugs, and how do we sequence these drugs to get the best effects? Critical. And this continued definition of the tumor and immune cell targets, the compartments that are affected, and the states of the cells in those compartments. Thank you.
Thank you very much, Dr. Balin. And I'm going to invite any questions, and I'm going to take the opportunity to actually ask the first question while people come to the microphone. So Dr. Sullivan, your group is among now many groups that have looked at the impact of entinostat on the myeloid compartment, on MDSCs, and on MMDSCs in particular. Your collaborators, Dr. Gabrilovich and the folks in melanoma, Liz Jaffe's lab has looked at this in pancreatic cancer, and it's been seen in breast cancer as well. So my question to you is, do you think that this is an entinostat or class 1 HDAC effect, or we know that all HDAC inhibitors are not the same? What is your feeling about the differences among the HDAC inhibitors and the families of HDACs?
Yeah, it's a great question. I think my knowledge about sort of the effects on that is mostly with entinostat and class 1 HDAC inhibitors. I think it is an effect that's related to that class of HDAC inhibitors. The data from the single dose phase 1 with entinostat is quite compelling, that with 1 dose you see reduction significantly in the majority of patients. So I think it is that effect.
Very quick comment. I agree. In that last paper I showed you, we took a look at a number of HDAC inhibitors, and in general, class 1 and 2 nuclear seem to be really the most important, but 3 is important, and in a couple of instances, HDAC 6 which has been used to potentiate therapy in multiple myeloma. So I think there's a big range of discoveries to go on about which of the HDACs and the families and how you have to target.
Wonderful. Any other questions from the audience? All right, so I'm going to thank Dr. Sullivan for his presentation.
I
and
have a question.
Oh, sorry.
For Dr. Sullivan. I'm just wondering, you looked at the CD8s in peripheral blood between responders and non-responders. Did you ever look at the proliferating CD8s, so Ki67 positive and CD8 positive?
It's a great question and very relevant. I'm just trying to remember all the other decks that we didn't include. I can't recall if we had that or not. A lot of the correlative data has actually been emerged over the last 4 to 6 weeks. Certainly, that's something that needs to be looked at, but I don't think we've looked at it quite yet, but thank you.
All right, wonderful. Thank you very much.
Thanks.
Next, I'd like to introduce Dr. Solange Peters from the University Center Hospital from Lausanne, Switzerland, who's going to be presenting her team's work, tumor mutational burden as a biomarker of survival in metastatic non-small cell lung cancer. Thank you very much.
Now it works. Thank you. Just waiting for the slides to start. Thank you. On behalf of my co-authors, I'd like to thank the scientific committee and the organizers for inviting us to present some MYSTIC data at the AACR 2019. The talk today will be entitled Tumor Mutational Burden as a Biomarker of Survival in Metastatic Non-Small Cell Lung Cancer, Blood and Tissue Tumor Mutational Burden Analysis from MYSTIC, a phase 3 study of first-line durvalumab plus or minus tremelimumab versus chemotherapy. Here are my disclosures. Durvalumab, an anti-PD-1 immunotherapy compound, is already approved for unresectable stage 3 non-small cell lung cancer and has shown also clinical activity in heavily pretreated patients with metastatic non-small cell lung cancer in the context of phase 2 and 3 previously presented trials. MYSTIC is a phase 3 open-label, multi-center study addressing the usual and classical population of naive, advanced metastatic non-small cell lung cancer characterized by the absence of EGFR mutation or ALK rearrangement with a performance status of 0 or 1. These patients were randomized between durvalumab and tremelimumab, or the classical regimen of platinum-based chemotherapy, and they were also stratified according to the percentage of tumor cells expressing PD-L1, with a threshold between positive and negative being used at 25%. There were 3 co-primary endpoints in MYSTIC. The 3 were focusing on the subgroup of patients with high PD-L1 as described by more than 25% of the tumor cell expressing PD-L1. And these 3 endpoints were overall survival of durvalumab versus chemotherapy, overall survival of durvalumab and tremelimumab versus chemotherapy, and progression-free survival of durvalumab plus tremelimumab versus chemotherapy. But today we will spend a little more time to look and envisage together the key exploratory endpoints of overall survival by blood tumor mutational burden and tissue tumor mutational burden in MYSTIC. The primary endpoint of overall survival has previously been presented during the ESMO immunotherapy meeting, and as you can see on these 2 graphs, this primary endpoint of superiority of durvalumab and tremelimumab versus chemotherapy in high PD-L1 expressors was not met. And as you can see here, both curves do not mean statistical significance, but maybe just a little, I would say, note for you is we consider potentially that durvalumab does bring a potentially clinically meaningful benefit in terms of survival with almost 3 months benefit and a hazard ratio of 0.76. But again, due to the statistical hypothesis and design in MYSTIC, this primary endpoint was not met and statistical significance for durvalumab or dual therapy was not met. What is the reason we consider that durvalumab and tremelimumab should potentially be combined, and how does it relate to TMB? So basically, high TMB might represent a surrogate of the presence of strong neoantigen, and some mutations might result in neoantigen release, leading to T-cell activation during the priming phase of the immune response in the lymphoid organs. However, would these T cells exist and travel back to the tumor? We know that PD-L1 expression leads to immune cell evasion, blocking this immune response. Tremelimumab, an anti-CTLA-4, is able to block this physiological mechanism and enhance the T-cell activation, the priming of T-cell and their expansion, which will travel back to the tumor, and at its role, durvalumab might reverse the immune suppression. That's the reason why durvalumab and tremelimumab can lead to anti-tumor activity via non-redundant pathways. What about TMB as a biomarker for immunotherapy? So TMB has shown potential as a predictive biomarker for immunotherapy benefit, and in all the available series today, independently of the PD-L1 expression. Across a wide variety of cancer types, higher tissue tumor mutational burden was associated with improved survival with immunotherapy. However, the overall survival benefit of immunotherapy versus chemotherapy or any other standard of care in high TMB patients was never demonstrated to date. We have been seeing this data of tissue TMB cutoff of 10 mutations per megabase being produced after the definition of this threshold for nivolumab and ipilimumab in the context of the phase 2 trial Checkmate 568. This threshold was defined by a plateauing of the response rate above these 10 mutations per megabase in the tissue. This threshold was used subsequently in the phase 3 trial Checkmate 227, where this high TMB threshold of more than 10 mutations per megabase was defining an improvement in progression-free survival for nivolumab plus ipilimumab versus chemotherapy. However, measuring tissue TMB, tissue tumor mutational burden, requires invasive acquisition, requires a biopsy with sufficient tissue, and subsequently a significant risk for a long turnaround time. We believe that blood TMB, measured from circulating DNA, might allow for a more rapid, less invasive testing, and biologically speaking, may also be more representative of the heterogeneity of metastatic lesions in the context of an advanced disease. In the MYSTIC trial, tissue TMB was evaluated using the same gene panel, the Foundation One tissue NGS platform from Foundation Medicine, the same which was used in the Checkmate 568 and Checkmate 227 I described previously. To be very short, it is a 315 gene panel representing a 1.1 megabase DNA footprint. Unfortunately, the data set for tissue TMB in MYSTIC trial only represent a small subset of the ITT population, 41%, corresponding to 460 patients in the MYSTIC trial. You can see the distribution of the availability, accessibility of the tissue TMB in the three arms of the treatment, quite well balanced, but a minority of the patients. Based on the fact that we have to keep in mind it's only a subgroup of the ITT population, the main patients' characteristics were quite similar between the ITT population and the tissue TMB evaluable population, but I have to make a note here with the exception of a slightly overrepresented proportion of squamous cell histology and smokers in the tissue TMB evaluable patient related to the imbalance of the small sample size. However, the most important here is to show that the outcome of the ITT population and the biomarker evaluable population, the tissue tumor mutational burden evaluable population, is strictly the same, with the same median overall survival in the three arms and the same landmark milestone, 24 months overall survival for the three arms, just allowing for the subsequent evaluations to be reliable on that regard. when you fix this threshold of 10 mutations per megabase for tissue TMB, you will, of course, define two different subgroups of patients. And you can see that they are quite similar for several patient characteristics, but they are different, and this is expected in the terms that there are more squamous cell histological subtype in the high TMB subgroup, and there are more smokers in the high TMB subgroup, as previously described in other series. Let's look at the overall survival data. The high TMB, tissue TMB subgroup was benefiting from durvalumab as compared to chemotherapy with a hazard ratio of 0.7. This hazard ratio was very similar to the one observed with durvalumab and tremelimumab with 0.72 as compared to chemotherapy. You can see also the milestone and the plateauing of the immunotherapy strategy at 24 months with 41.7% of patients being survivors for D plus T, durvalumab and tremelimumab, versus 35 for durva versus 22.7 for chemo. When you look at the subgroup of patients with a lower number of mutations than 10, this phenomenon is not observed. And if anything should be potentially stressed about this hazard ratio all above 1, it's potentially that chemotherapy represents a better option in this patient population. Blood tumor mutational burden was evaluated in MYSTIC for the first time with a description of the Guardant Omni sequencing platform for Guardant Health, which comprises a 500 gene panel with a quite equivalent 1 megabase DNA footprint. It's the first time it was used, so I just described that this Guardant OME blood TMB algorithm incorporates somatic single nucleotide variants and small insertion deletions, and interestingly accounts for low tumor shedding and low ctDNA input too. This is the largest blood TMB data set ever presented, and in MYSTIC it represented 72.4% of the ITT population, representing 809 patients. These patients, as you can see on the bottom of this slide, were equally distributed in the three randomized treatment arms. Do we find a correspondence between these two biomarkers, tissue and blood TMB? So it's very important to look at this curve with a little limitation. Only 31.5% of the ITT population in MYSTIC had matched available specimen for tissue TMB and blood TMB reading. But taking this limitation of small number into account, we could find a positive correlation between tissue and blood TMB using the two technologies described, with a Spearman's of 0.6 and a Pearson's of 0.7. If you look at this, the characteristic of blood TMB biomarker evaluable population and ITT population of MYSTIC, now it's a big group of patients, and no difference in the main patient characteristic was observed in these two cohorts of patients. And equally as before, when you look at the outcome in these two populations of patients, the intent to treat and the biomarker evaluable population, the BTMB evaluable population, the median overall survival in the three arms and the landmark survival were the same, allowing us to go for subsequent analysis. This is the very first preliminary exploratory analysis using a threshold of 16 mutations per megabase, which was presented at the ESMO IO last year. It was really the first kind of deciphering of blood TMB from MYSTIC. And you can see here in this graph that, interestingly, what was striking was the benefit in overall survival for durvalumab and tremelimumab versus chemotherapy with a hazard ratio of 0.62. To a lower extent, in a weaker fashion, durvalumab was also showing some benefit versus chemotherapy with a hazard ratio of 0.8, but still a slight difference was visible between the dual immunotherapy, and only a small benefit was visible for durvalumab versus chemotherapy. And you can see here the 39.1, 29.6, and 18.2, 24 months overall survival. Starting from that point, we decided to try to better understand the optimal outcomes for the patient benefit, of course, but also the potential contribution of durvalumab monotherapy and on tremelimumab added to durvalumab, and to do so to further explore some potential other thresholds of TMB, blood TMB in MYSTIC. And here you can see the various thresholds which can be used for blood TMB in MYSTIC. And you can see in this graph that when you look at durvalumab, survival benefit is observed in all subgroups of patients defined by a high TMB. So all thresholds were equally representing a benefit, and across all the thresholds, durvalumab was showing this benefit. I would even say irrespective of the threshold chosen for defining high TMB. When you look at durvalumab and tremelimumab, again, all the patients defined by a high TMB according to the thresholds defined were showing a benefit in overall survival as compared to chemotherapy. However, the magnitude of this benefit was improved with increased number of mutations and an increased threshold for blood TMB. Importantly, too, if you look at the hazard ratio and the reference intervals, this data suggests that the benefit of durvalumab and tremelimumab, the activity, is higher than durvalumab monotherapy over chemotherapy. And based on that, we decided to move with a more or equal to 20 mutation per megabase cutoff to conduct a subsequent analysis based on the observed specific effect of durvalumab plus tremelimumab in this patient population and the magnitude of this effect in this population. So now we look at the population of patients that you divide by more or less than 20 mutations per megabase in the blood, and again you will find a difference that was expected. There will be more smokers in the patient population with more than 20 mutations per megabase in the blood and more squamous histology. And here are the survival results. So that time you will observe in the patients with a high BTMB of more or equal to 20 mutations per megabase, you will observe a benefit in survival of durvalumab with a hazard ratio of 0.72. But with a higher magnitude, to a broader extent, you will observe a very significant benefit of durvalumab plus tremelimumab looking at survival versus chemotherapy with a hazard ratio of 0.49. Again, and very interestingly here, you will see that durvalumab plus tremelimumab with this threshold is better than durvalumab with a difference versus durvalumab and a hazard ratio of 0.74. And this is very well represented on the tailoring of the curve, the plateauing of the curve, when you can see the 2 years overall survival of 48.1% for durvalumab and tremelimumab, 33.8% for durvalumab monotherapy, and 19.4% for chemotherapy. Interestingly, this was again not observed with quite equivalent results in the subgroup of patients with a lower number of mutations, less than 20 mutations per megabase, and if anything had to be potentially stressed is that chemotherapy is still a good option in this patient population. Very importantly, looking at optimization of patient outcomes, the other endpoint we all want to look at is PFS. Using this threshold of more than 20 mutations per megabase in the blood, you will see here that the PFS is improved under durvalumab versus chemotherapy with a hazard ratio of 0.77. Again, with a higher magnitude and to a wider, larger extent, the PFS is even more improved with durvalumab plus tremelimumab with a hazard ratio of 0.53 as compared to chemotherapy. And a significant difference was observed for durvalumab plus tremelimumab versus monotherapy durvalumab with a hazard ratio of 0.76. And here, the milestone analysis at 1 year of progression-free survival, the percentage of patients being progression-free, is very striking with a plateauing of the immunotherapy strategies, 38.6 for dual inhibition, 30.3 for monotherapy, down to 2.3 for chemotherapy in this patient population. Let's look at the PFS for patients with less than 20 mutations per megabase. And here again, chemotherapy is probably better, and in particular in the first months of treatment, looks like being more relevant for this patient population as compared to any of the options with immunotherapy. Again, patient benefit is also about response rate, and here we can mimic what we just presented with the previous endpoints. The overall response rate was strongly improved with this threshold of 20 mutations per megabase for durvalumab plus tremelimumab, 48.4%, down to 29.9% for durvalumab and 21.4% for chemotherapy. And again, under this threshold, it looked like chemotherapy was offering a better response rate with 31.4% as compared to 20 and less for the immunotherapy strategy. Once a response was met, was hit using this treatment, it's very important to look at how immunotherapy can change the duration of treatment. And the patient remaining in response at 12 months was way better by using immunotherapy as compared to using chemotherapy. And you can see particularly when you look at high TMB subgroups, this difference is even stronger, with 80.3% remaining in response at 12 months with durvalumab, 81.7% with durvalumab plus tremelimumab versus 7.2% for chemotherapy. But this phenomenon was seen in both TMB subgroups, but again with a reinforced effect in the patients with a higher blood TMB threshold being the selection criteria. PD-L1 and blood TMB remain in MYSTIC-independent biomarkers. I told it in the introduction, most previous series tended or were able to show that PD-L1 and TMB are not correlated. And you can see here on the left-hand side plot that BTMB values did not correlate at all with PD-L1 expression at any threshold for PD-L1 or blood TMB, with a Spearman's of 0.05 and a Pearson's of 0.01. And looking at the Venn diagram on the right-hand side, it's very important to look at the positivity as defined by MYSTIC and to also conclude together how different are the patient population you are describing if you describe positive population based on PD-L1 or positive population based on blood TMB, with only 9% of the patients meeting both positivity and both criteria. So you really look at different patients when you envisage these biomarkers. Last but not least, are we also defining a patient population which, besides the clinical benefit, will also experience a different pattern of toxicity and potentially different safety issues? So here are the data about the overall safety set in MYSTIC and the blood TMB subgroup of more than 20 mutations per megabase. And you can focus on any of these parameters, for example, treatment-related adverse event or high-grade treatment-related adverse event. There's strictly no difference in the toxicity patterns that this subgroup of patients will experience as compared to the overall safety set in the MYSTIC trial. With this, I go to my conclusion. This exploratory analysis of MYSTIC is showing that TMB, tumor mutational burden, was predictive of the overall survival benefit with first-line durvalumab and first-line durvalumab and tremelimumab in metastatic non-small cell lung cancer. To go into the details, in tissue tumor mutational burden or more or equal than 10 mutations per megabase population, the overall survival was improved with durvalumab with or without tremelimumab versus chemotherapy. However, a note of caution, the small data set of tissue TMB available samples and patients will limit this interpretation in the future. Hazard ratio for overall survival was improved with increasing blood TMB levels for specifically the durvalumab and tremelimumab arm versus chemotherapy. We have been defining this threshold of 20 mutations per megabase in the blood, and this threshold was associated with an improved overall survival of durvalumab versus chemotherapy, with a hazard ratio of 0.72 and a 24-month overall survival rate of 33.8 versus 19.4. And we also have been describing this specific and markedly improved overall survival and the greater clinical benefit when durvalumab is combined with tremelimumab versus chemotherapy, indicating the potential contribution of this component of tremelimumab in this specific setting, with a hazard ratio for overall survival at 0.49 and a 24-month overall survival rate strongly improved from 48.1 to 19.4 for chemotherapy. Also a PFS hazard ratio of 0.53 and an improvement in overall response rate from 48.4 to 21.4 for chemotherapy. We do believe that based on this exploratory analysis, further investigation as prospective validation of blood TMB as a predictive biomarker for immunotherapy is warranted and should potentially be evaluated in as many as possible future clinical trials looking at immunotherapy across various solid tumors. With this, of course, all the authors of MYSTIC would like to warmly thank the patients, the families, and the caregivers. We also want to thank all the investigators and site personnel from 203 sites in 17 countries, and I'd like to thank the medical writing support from Ashfield Company and the collaboration with AstraZeneca in order to generate with my colleague the data I presented to date. This is the list of MYSTIC investigators, and last but not least, I'd like to thank you all for your very kind attention.
Thank you very much, Dr. Peters. And now I'd like to introduce Dr. David Gandhara, who is here from the University of California, Davis Comprehensive Cancer Center, to share his perspective on this trial based on his expertise as a drug developer and an expert in non-small cell lung cancer. Thank you.
So it says Peters Gandhara. Do you know, is that? I see. I want to let you know this is cutting into my 10 minutes of discussion time.
We're going to restart the clock, don't worry.
Fantastic. Too simple. Well, thank you very much. Thanks for the opportunity of discussing this interesting paper in this session dedicated to Ki Hong, very appropriate, a mentor and a friend to many in this audience. You can see I've titled this discussion Mystic Search for a Predictive Biomarker with a subtitle of Is the Glass Half Full? Since Dr. Peters and I are both enophiles, you'll see soon the meaning of this. My disclosures. And I start with this slide, which shows the results of checkpoint inhibitor therapy in the first line in advanced non-small cell lung cancer, many trials. You see that only 2 were negative for the primary endpoint. And I'm sorry I'm advancing, but nothing is happening. I'll advance from the computer. The Checkmate 026 and the MYSTIC study, which was presented today. Well, what do these 2 trials have in common? Both were phase 3 trials in first-line checkpoint inhibitor therapy. Both were randomized against platinum chemotherapy. Both were statistically negative for primary endpoints. And both identified populations within the studies who benefited from therapy based on tumor mutational burden subset analysis. So I raise the question here, is the glass half full? In these 2 very well done, very well conducted studies, does the finding of mutational burden, whether in tissue or in blood, help us in our understanding of immune response? And does it move the field forward? Now, I point out that if you look at the glass half empty, it looks pretty much like the glass half full. But it is connotation that makes us view these differently, and it is connotation that I will talk about today. I start with this slide from the Cancer Gene Genome Atlas project. Not only to show quantitatively that lung cancer has the highest mutational burden among many tumors for mutations per megabase, but also qualitatively that it is different. These orange mutations are C to A transversions. They are typically associated with tobacco carcinogenesis, and they are among the most neoantigenic, and you can see they are concentrated within non-small cell lung cancers. So what about mutational burden as a predictive biomarker? We know now we can measure tumor mutational burden by many techniques, some research techniques such as whole exome sequencing, or more recently, comprehensive genomic profiling by next-generation sequencing, with a variety of tests, some of which are shown on this slide. Some of these, such as the foundation medicine, as you can see in the upper right, have been associated very closely with whole exome sequencing, or perhaps more important, with predicted neoantigen load. I'll come back to this in a few moments. And then, as you can see in the diagram below, and as Dr. Peters demonstrated in her study, and essentially all the studies done in lung cancer, TMB and PD-L1 identify largely non-overlapping populations. The potential importance of TMB, as I mentioned, was first brought to the lung cancer community attention in this checkmate trial, 026, randomizing patients to nivolumab or platinum chemotherapy. And as you can see, for the primary endpoint based on PD-L1, it was a negative study. But I think to the credit of the investigators, including Dr. Cabone and Dr. Peters, they showed that for a group of patients identified by tumor mutational burden by whole exome sequencing, there was benefit, as you can see on the left, those patients with high tumor mutational burden benefiting from nivolumab. But conversely, it is the green chemotherapy Kaplan-Meier curve on the right. They also, I think of significant importance, showed that the whole exome sequencing data correlated well with the gene set found within the Foundation 1 panel. And this set the stage for further evaluation of this test. And I think interesting to this presentation today, this study showed that patients who were high, that is, patients who had both high PD-L1 and high TMB, were the patients most likely to benefit from nivolumab, not seen in the chemotherapy arm. So these set the stage then for this presentation that we made at ASCO last year, looking across approximately 1,000 patients treated with atezolizumab across multiple tumor types, although primarily lung cancer and bladder cancer, multiple lines of therapy. As you can see on the left, this is the assay which has correlated well with neoantigen load, and you can see that it is also equally predictive of response rate to atezolizumab, as was neoantigen load. Now, in this analysis, we identified a TMB in tissue of greater than or equal to 16 as our best cut point associated with response, duration of response, progression-free survival. Now, we could have, of course, gone to a higher TMB, but as you see, you lose power in this analysis with 1,000 patients when doing so. Well, that was with monotherapy. What about going to combination immunotherapies such as nivolumab and ipilimumab, where TMB by the Foundation Medicine assay was a co-primary endpoint? And as you can see, this was a positive study for progression-free survival using a TMB cut point of greater than or equal to 10. As you can see on the right, this was derived from a retrospective analysis of this combination in 98 patients. I will state that this is not derived, of course, from the tens of thousands of patients that went into derivation of the cut point for Foundation Medicine. Most importantly in this study, those patients with absent PD-L1 expression but high TMB at a level of 10 had improved progression-free survival. And conversely, those patients who were low had no benefit over chemotherapy for PFS. Now, those results were in tissue. Moving now to blood, last year we published the analytical and clinical validation of tumor mutational in blood, derived from the Foundation Medicine assay, but with a little different computational methodology. Again, tumor mutational burden greater than or equal to 16 was selected as the best endpoint, realizing this is a continuous variable. You can pick a higher endpoint, but of course it is a smaller percentage of the population. And this correlated, you can see, with progression-free survival, also with response rate, in an agreement analysis where we took identical samples from cell-free DNA and we ran them through both computational algorithms, the tissue foundation, the blood foundation, you can see there was excellent correlation with the Spearmans of 0.93. And as others have shown, we show that there was limited overlap between PD-L1 high and TMB blood high in this analysis. But importantly, although a small group of patients, if patients were high, the PFS hazard ratio was 0.38, overall survival 0.23. So perhaps supporting a combination analysis. Prospectively, our results have been taken into a proof of principle trial, B first. It has been presented at ESMO last year by Dr. Kim and by Dr. Valchetti earlier at ASCO. And as you can see, using this cut point of greater than or equal to 16 as a selection factor for atezolizumab, there are increasing response rates in TMB 10, 16, and 20. But as you can see, once you get to 20 in this small study, it is a very small group of patients. And this is being taken, of course, now into phase 3 with a trial that is ongoing, the BFAST trial, to clinically and from a regulatory standpoint validate blood TMB by this assay. Well, let's return to MYSTIC. With that perspective then, these are the data that were presented by Dr. Rizvi at ESMO IO last December. Now, Dr. Peters presents these data at a cut point of greater than or equal to 20, and she has elegantly explained the rationale for this, and my thanks to her for not only providing the slides, but for really setting the stage for this discussion. She described why the checkpoint of 20 was selected for further evaluation based primarily on the combination data with D plus T. I will point out that as we observed, this is a shrinking denominator phenomena. As you can see, as you go from 16 to 20 is a smaller percentage of the population. So if adopted, the question would be, are we restricting the use of a treatment to a very select population? Now, this assay, the Omni assay by Guardant, has not been fully published, but there are multiple presentations on it, and it is a robust assay, as Dr. Peters described, and as you can see from the in silico subset matching, that it compares favorably with whole exome sequencing. Dr. Peters also presented the correlation of blood TMB with tissue TMB and the association with efficacy outcomes at a level of 10. So I return to my question, is the glass half full, and what would it take to fill up that glass of red wine? Well, we'd like to know more about TMB as a predictive biomarker within this study, more about the methods and of the assay and the analysis. This will be presented at a future meeting. We'd like to know more about these cut point associations and the association with efficacy. Why? Because the goal is to maximize the potential predictive benefit, but also maximize the percentage of patients who would be eligible for therapy. We also want to know about the combination data. High, low for PD-L1 and TMB. Why? Because this may be, at the present time, our best combination biomarker for checkpoint immunotherapy. And lastly, we want to know how these data harmonize with other TMB data using other assays, checkpoint immunotherapy monotherapy, IO combinations, checkpoint with chemotherapy. These may well be different. Luckily, there are harmonization assays ongoing, such as this one led by the Friends of Cancer Research. There is also one in Europe. Why do we need these harmonization assays? Because it is estimated that there are over 2,000 ongoing trials of checkpoint immunotherapy requiring 380,000 patients. This is accrual fatigue. We need to address this unmet need for clinical validation of predictive biomarkers, and this is an opportunity. Thank you.
Thank you very much. I think we have time for maybe one or two questions. Dr. Herbst?
Yes, very nice presentation and discussion. I'm wondering what's in the glass. You've told us it's wine, but what is the biology here? These patients that are responding quite well, have you looked at the tumor microenvironment? Has there been any exploration for specific neoantigens? That might be helpful to sort of figure this out as well.
Tumor microenvironment, it's going to be looked at.
Yeah, so these data are ongoing, trying to look at further biomarker in MYSTIC. You've seen that one of the limitations in the MYSTIC trial will be the tissue analysis in general, because only 10 slides were asked for, and after PD-L1 and after the TMB, not so much is remaining. So everything which might be about looking at circulating immune signature might still be performed. As in the future, of course, other types of biomarker will have to be looked at. I agree with you with the idea of a prediction of neoantigens being the next step further down the road of this surrogate, which is TMB, and kind of innocently counting mutations. But you also have been looking, like me, at all these algorithm and predictive neoantigens facilities that are accessible to you, and none is really reliable in the way they might really be used as a biomarker for any subsequent immunotherapy. So I think the field of the next subsequent trial is to validate this TMB story before we will be able to use any other biomarkers beyond PD-L1, to my opinion.
I would just add that a very important study in this regard, I think, is INSIGNA, which is a joint ECOG, SWOG, NCI funded trial in first-line therapy, which I think will start this next month. And embedded within it is a very nice correlative science component to establish immunosignatures, which will clearly include the microenvironment. And I will just say that it's hard to hear up here. It's a little muffled, so you can...
I think just one more. Please go ahead.
Hi, Dave Rim from Yale. I wonder if you showed very nicely that the higher you get with TMB, the more predictive or the greater the predictive power. What happens if you switch them around? Because for tissue TMB, you use 10, but then for blood TMB, you use 16. What happens if you would have used 10 in the blood and 16 in the tissue, and would you have seen a switch in the results?
Well, you've been seeing how blood TMB and tissue TMB to a certain extent correlate, right? So the higher you go for tissue TMB, the higher you go for blood TMB. So you're proposing that we accept in the blood to go to a lower threshold of mutation. So to my opinion, the predictive ability of this new threshold you define, and like David has been showing in this Nature Medicine paper, might make you come down to a hazard ratio which will be less significantly in favor of the immunotherapy arm. So I think both tissue and blood move in parallel with some different threshold based on the gene panels we use and the sensitivity threshold we use. But they move in parallel. The problem with tissue is, based on the limitation we have to accessibility to tissue, we are not able to test so many of these thresholds. But in the blood, you can do that. So I think we will be more flexible with the blood. But again, they move in parallel. So I would not twist them one against the other one, because you will lose your predictive ability. Did I understand well?
Yeah. I'm sure Dr. Peters will be around after the session to answer any further questions as well. And so I'm going to move forward with our next presentation, which is Dr. Julia Strauss, who is presenting an exciting new novel agent, a phase 1 evaluation of M7824, which is a bifunctional antibody that targets TGF-beta and PD-L1, and this is in patients that have HPV-positive associated cancers. And thank you very much to our previous discussant and presenter.
Double click. I'm trying to do it. Sorry. Now you have a lot of practice. Yeah. Oh, we have to do it over here. Sorry. On behalf of the authors, I would like to thank the conference organizers for the opportunity to share data with you today on the phase 1 evaluation of bintrafusp alpha, otherwise known as M7824, a bifunctional fusion protein targeting PD-L1 as well as TGF-beta in patients with HPV-associated malignancies. By way of background, HPV is the leading cause of oropharyngeal, anogenital, and cervical cancers in the US and worldwide. Each year, more than 30,000 patients are diagnosed with HPV-related malignancies in the US, and more than 630,000 patients are diagnosed with HPV-related malignancies worldwide. Thus, this patient population is in urgent need of effective therapies. And unfortunately, traditional chemotherapies offer only modest improvement in survival outcomes for these patients. As with other solid tumors, the PD-L1 pathway is significantly important in these cancers in leading to disease progression and immune tolerance. Studies have shown that the gene encoding for PD-L1 is often amplified in HPV-positive malignancies, including in cervical and vulvar cancers. In addition, PD-L1 is often highly expressed in these cancers, including in HPV-positive squamous cell carcinomas of the head and neck. In addition, in earlier stages of this disease, including in cervical intraepithelial neoplasia, higher PD-1 and PD-L1 expression in the tumor microenvironment has been shown to correlate with higher grades of disease and worse prognosis. Therefore, PD-L1 inhibition and PD-1 inhibition is critically important in patients with HPV-associated malignancies. Studies targeting and inhibiting PD-1 to date in cervical, anal, and HPV-positive squamous cell head and neck carcinomas have produced response rates ranging from 13 to 24%. Many of these studies have led to FDA approval of nivolumab and pembrolizumab for squamous cell head and neck carcinomas, as well as pembrolizumab for PD-L1 positive cervical carcinomas. In addition to the PD-L1 pathway, the TGF-beta pathway is also critically important for patients with HPV-associated malignancies. TGF-beta plays an important role in tumor growth, progression, metastasis, and resistance to therapy. It leads to tumor angiogenesis. It drives epithelial to mesenchymal transition, and it suppresses the immune system, causing immune tolerance via a number of methods, including it impairs cytotoxic T cell and NK immune responses. It impairs T cell infiltration into the tumor microenvironment. It drives the recruitment of M2 macrophages and MDSCs into the tumor microenvironment and promotes Treg function. In addition, a number of large genome-wide association studies have shown the TGF-beta pathway to be significantly associated with HPV-positive head and neck squamous cell carcinoma, as well as cervical carcinoma, and that TGF-beta receptor 1 is significantly overexpressed on these tumor types as compared to benign tissue. This strongly suggests that dysregulation of the TGF-beta pathway may play a critical role in HPV-mediated carcinogenesis, making this pathway a potential therapeutic target for these diseases. Bintrafusp alpha, otherwise known as M7824, is a bifunctional fusion protein composed of the extracellular domain of TGF-beta receptor 2 fused to the human IgG1 monoclonal antibody and inhibitor of PD-L1. In a phase 1 study of bintrafusp alpha in patients with advanced solid tumors, bintrafusp alpha was found to be generally well tolerated and produced durable responses in several tumor types, including in patients with HPV-positive head and neck squamous cell carcinoma, as well as cervical carcinomas. The data that I will present to you today will be a post hoc analysis of patients with HPV-associated malignancies which were enrolled on the larger phase 1 trial of bintrafusp alpha in patients with advanced solid tumors. It will include 17 patients with HPV-associated malignancies which were enrolled on the dose escalation component of the phase 1 trial, where patients received escalating doses of bintrafusp alpha ranging from 0.3 to 30 mg per kg every 2 weeks. as well as 26 patients with HPV-associated malignancies enrolled at 20 worldwide sites on the expansion phase and received the recommended phase 2 dose of bintrafusp alfa of 1,200 milligrams every 2 weeks. In all, this data on 43 patients with HPV-associated malignancies will include safety and efficacy data. Data will also be presented on 36 patients which had HPV-positive confirmed disease. This disease was confirmed in the dose escalation component of the trial via a DNA PCR-based assay, the Cobos 4800, which is FDA approved for HPV detection, as well as RNA sequencing confirmation, which was done on the expansion cohorts of this trial. Inclusion criteria for patients on the phase 1 overall larger phase 1 trial included patients with metastatic and locally advanced disease who were unselected for HPV status as well as PD-L1 expression. In addition, these patients had measurable disease per RECIST, and patients had not had prior checkpoint therapy. This slide shows the demographic data of these patients. 2/3 of patients were female with a median age of 56. Slightly over the majority of patients had cervical carcinomas. About 1/3 of patients had squamous cell carcinomas of the head and neck, and 9 to 10% of patients had anal carcinomas. 2/3 of patients had received 2 or more lines of systemic therapy for advanced cancer prior to enrollment. Predominantly 88% receiving traditional cytotoxic chemotherapies, with 60% receiving monoclonal antibodies, including cetuximab and Avastin, as per their disease state. Only 3 patients, or 7%, have previously received immunotherapy. In all 3 patients, this was adoptive T-cell therapy. No patients have received prior checkpoint therapy prior to enrolling. As previously mentioned, 36 out of 43 patients had HPV confirmation, which was 84% of patients. 3 patients, or 7%, were found to be HPV negative on confirmation, and 4 patients had an unknown status. On the left side of this slide, you can see the table which represents the treatment-related adverse events for the overall 43 patient population. This includes individual AEs of any grade that occurred in more than 10% of patients, as well as grade 3 or greater AEs that occurred in any patient. Autoimmune toxicity, which was observed with this agent, was similar to what has been described with traditional PD-1 and PD-L1, anti-PD-1 and anti-PD-L1 therapies. The most common adverse events occurring in more than 10% of patients were dermatologic AEs, such as rash and pruritus, and hypothyroidism. 16% of patients discontinued treatment for treatment-related AEs, the most common of which was autoimmune toxicity. When autoimmune toxicity did occur, it was well managed by holding treatment and offering steroid taper per selected cases. The only grade 4 AE, which was treatment-related AE, which was observed, was isolated asymptomatic hypokalemia in the context of gastroparesis, which was attributed to bintrafusp alfa. No treatment-related deaths were observed. 2 additional toxicities were observed. In 20% of patients developed keratoacanthomas, or low-grade squamous cell carcinomas of the skin, which has been described with other TGF-beta inhibitors. These lesions were generally well managed, either with observation or local therapy, and did not require treatment discontinuation. In addition, low-grade mucosal bleeding was also observed in patients. This has also been observed in the literature with other TGF-beta inhibitors. These examples of this included gingival bleeding and epistaxis. And this is thought to be due to vascular fragility based upon normal platelet aggregation tests, as well as coagulation workup on a number of patients. The confirmed, in the overall larger 43 patient population, the confirmed objective response rate per RECIST 1.1 was 27.9%. In patients with HPV-positive confirmed disease, the confirmed objective response rate per RECIST was 30.6%. This included 2 patients with cervical carcinoma who had complete responses, as well as 4 patients with squamous cell carcinomas of the head and neck with durable PRs, 4 patients with cervical carcinoma with durable PRs, 2 patients with anal cancer with PRs. In addition to these responses, 3 additional patients achieved delayed but durable partial responses after initial disease progression. Including these responses, the total response rate of the overall 43 patient population was notably 34.9%, and the total response rate of the HPV positive confirmed population was notably 38.9%. The responses which were observed were durable and ranged from 2 to ongoing at 25 months. The median duration of the response was not reached as of the data cutoff. 9 of 12 of the responses per RECIST were still ongoing as of the data cutoff, and including patients who had delayed but durable partial responses, 11 of 15, or 73% of the responses, were still ongoing as of the data cutoff. On this slide, you can see the waterfall plot for the larger 43 patient population broken down by tumor type. Notably, 21 of 43 patients, or nearly 50% of patients, had some disease reduction with treatment. Disease reduction was seen in several patients who had visceral metastasis. Included on this slide are 2 examples. On the left is a patient with anal carcinoma with mets to the liver, and on the right is a patient with cervical carcinoma with mets to the lung. If you look at the bottom half of this slide, at the images at the bottom half, you can see that in both cases, in both patients, had near resolution of disease by 3 months on treatment. PD-L1 expression was also evaluated in patients enrolled in the expansion cohort. This includes patients with cervical cancer, which are seen in purple on the left side of the slide, as well as patients with squamous cell carcinoma of the head and neck, which are seen on the right side of the slide in orange. What you're seeing is the best overall responses for these patients. In green, you can see the PD-L1 expression by IHC. Notably, responses were observed regardless of PD-L1 expression. On this slide, you can see the Kaplan-Meier curve for overall survival. The median overall survival for patients on the overall cohort of 43 patients was 16.2 months, and the median overall survival for patients in the HPV-positive confirmed group was not reached after a median follow-up of 16.6 months. Although it is not fair to compare across trials, to put this data in context, similar studies in similar disease populations evaluating anti-PD-L1 and anti-PD-1 therapy have shown median overall survivals ranging from 9.1 to 11.5 months. And it is additionally important to note the tail on the curve, which extends out beyond 18 months. In conclusion, in patients with treatment-refractory HPV-associated malignancies, bintrafusp alpha led to response rates that compared favorably with traditional anti-PD-1 inhibitors, with a historical objective response rate of 13 to 24%. Per RECIST version 1.1, confirmed objective responses were seen in 27.9% of patients on the overall 43 patient cohort and 30.6% in patients with HPV-positive confirmed disease, including the 3 patients who had delayed but durable responses after initial disease progression. The total response rate on the overall cohort was 34.9%, and most notably was 38.9% in patients with HPV-confirmed disease. The responses, again, were durable, with, at the time of the cutoff, 4 patients having durations of response exceeding 18 months, and 11 of 15 responses still ongoing. Responses to bintrafusp alpha occurred irrespective of tumor type or PD-L1 expression. and the safety profile, at least as in regards to autoimmune toxicity, was similar to that has been seen with other anti-PD-1 and anti-PD-1 therapies. There was the observation of keratoacanthomas as well as low-grade mucosal bleeding, which has been previously described in the literature with other TGF-beta inhibiting agents. Therefore, dual blockade of TGF-beta as well as PD-L1 with bintrafusp alpha represents a promising therapy for patients with HPV-associated malignancies and continues to be evaluated in this patient population in an ongoing phase 2 trial at the NCI. On behalf of the authors, I would like to thank the patients and their families, as well as the investigators who contributed to this study. This study was funded in part by the Center for Cancer Research, National Cancer Institute, as well as in part by Merck KGaA of Darmstadt, Germany, as part of an alliance with GlaxoSmithKline. Medical writing support was provided by Clinical Thinking and also funded in part by Merck KGaA, as well as GlaxoSmithKline. I'm happy to take any questions.
Thank you very much, Dr. Strauss. And now I have the pleasure of introducing Dr. Moa Gillison, who left us at Johns Hopkins for the eventual warmer pastures of MD Anderson, and has spent her career really focusing on the impact of HPV on malignancy. We look forward to your discussion.
We're down here to yourself and then start. Okay, thank you. This slide is definitely wrong. I'll show my disclosures that are accurate in a minute. It's an honor to present here in Waun Ki-hong's memorial session because he was kind of a guardian angel of my career from a distance, very supportive of me. These are my accurate disclosures. These are the companies that I've consulted for over my career. I think it's best to list over my career. And this includes EMD Serono, which is the company that manufactures this drug. As we've heard, approximately 5% of the world's tumor burden each year is attributable to a single necessary cause, which is HPV infection. And despite one of the most important advances in cancer prevention research, which is the development of prophylactic HPV vaccines, the incidence of these cancers increases each year. Just due to population growth, it's expected by 2030 that there will be 700,000 cervical cancers alone diagnosed each year worldwide. In the United States, these cancers also continue to increase in incidence each year. The burden increases largely because of increases in HPV-positive oropharynx cancer. There were only about 26,000 HPV-positive cancers diagnosed each year in the United States as of 1999. And each time the CDC releases their new data, I update this slide and the numbers continue to go up. And as of 2015, there are about 34,000 HPV-positive cancers diagnosed in the US. As you can see by the SEER 5-year OS data from these cancers, is that the majority of patients with local regionally advanced disease who have developed refractoriness to radiation and cisplatin will go on to do quite poorly. And historically, there hadn't been any therapeutics that were active in that patient population with platinum refractory disease. But over the last couple of years, we now have 3 different approvals in this setting for agents that target the PD-1 or PD-L1 pathway. And this represents the response rates you see in those various trials specific to the HPV-positive subset. And the overall response rates range from about 14% to 26%. So we heard today about some potential promising new activity in this space that appears to potentially increase activity over those response rates that we saw with PD-1 or PD-L1 targeting alone. Although those of us who are in the field were extremely excited to have a new class of drugs, we were also quite realistic about how the majority of patients progress rapidly and only a small proportion of patients benefit. So clearly, there's room for improvement. And here we're seeing some preliminary data that perhaps targeting the TGF-beta pathway in addition to PD-1 or PD-L1 may lead to improvement. But I want to have some caution with regard to just focusing on the overall response rate, because I don't think that's the important signal we're seeing here. Overall, the response rate is still within the confidence bounds of the prior studies, and the results in prior studies fall within the confidence bounds here. So it wasn't really the overall response rate that struck me with this data. They importantly looked at some potential that the overall response rate wasn't just driven by a high PD-L1 proportion. We have shown previously that your response rate to these agents increases as a function of the proportion of cells of the tumor that have PD-L1 expression, so that response rates for patients with PD-L1 greater than 10% will be in the 30% range, and above that, even higher. So I applaud their having looked to see that there were responders in the PD-L1 less than 1% group, but there still remains to be further data with regard to, for instance, what we just heard, TMB, whether or not this is confounding by other factors that we already know will increase response rates to PD-1 and PD-L1 targeting populations. But what I was quite struck with in this data was really the deep responses and their duration. The median duration of response in most trials for these cancers is on average about 7 months. And in this data, you see the duration of the response is quite compelling. And I think that might be what's driving this even more compelling overall survival curves with a higher median OS in this patient population than we've seen in the other trials. And striking to me is the 12-month OS that we see is generally approximately 36%. So going up to as high as potentially 62%. I think it's the duration of the response and its impact on OS that I find most intriguing about this agent. A word about toxicity. I think that we did see here a higher rate of grade 3 or greater toxicities than we see on average with PD-1 targeting alone. The treatment discontinuation rate of 16% was higher than we've seen in the trials with PD-1 alone of about 6%. The dermatologic rash events were markedly higher, 8% with PD-1 targeting alone, and rash, pruritus, dermatitis, and skin lesions were 20 to 25% each in this patient population. And that's due to basically a pharmacodynamic on-target effect. By targeting TGF-beta and its known role in epithelial differentiation, controlling proliferation, and wound healing. Just a brief statement about keratoacanthomas. They're low-grade, considered largely benign low-grade tumors, and they arise rapidly and not in the setting of prolonged TGF-beta inhibition. They tend to regress on their own. Whether or not that natural history has changed for people who are on these agents is something that will be important to follow. And they can be difficult to differentiate from squamous cell carcinomas. So important to follow that finding up. And this is consistent with a known familial syndrome of keratoacanthomas in patients who have mutations in the TGF-beta R1 receptor. They're also seen in other drugs, including BRAF inhibitors and imiquimod and other TGF-beta inhibitors. So why might it make sense that targeting TGF-beta may be important in HPV-associated malignancies? Well, in vitro data and more importantly, clinical data directly on patients with cervical dysplasia show that the TGF-beta pathway is disrupted during the histopathologic progression of cervical cancer. Early on, TGF-beta appears to be anti-carcinogenic. TGF-beta itself, when E6 and E7 are expressed at low levels, can suppress E6 and E7 expression, and it can decrease proliferation, increase differentiation, and be pro-apoptotic in early stages. But later in the development of carcinoma in situ and invasive cancers, you see actually with higher E6, E7 expression, those proteins can actually bind to the TGF-beta promoter and increase TGF-beta expression by the tumor, increasing immunosuppression in the tumor environment, angiogenesis and metastasis, and genomic instability. We were surprised to find in our recent publication that many of the pathways that are altered by somatic mutation and deletion in HPV-positive head and neck cancer are already targeted for disruption by the E6, E7, and E5 viral proteins, oncoproteins, and the TGF-beta pathway was one of those pathways. Here's it represented as the TGF-beta pathway, which TGF-beta binds to the TGF-beta R2 receptor, activates the R1 receptor, signals through SMAD, and then translocates into the nucleus and drives expression by recruiting other transcription factors to the DNA, including EP300. Well, E7 can bind SMADs in the nucleus and prevent that transcriptional activation. E5 can prevent SMAD4 translocating to the nucleus. E6 and E7 can increase the expression of a negative regulatory SMAD7. And then in both cervical cancers and head and neck cancers, we see frequent mutation and deletion in the pathway, including of the receptors of SMAD4 and of these transcriptional co-regulators, in particular EP300. So what we see is the canonical TGF-beta pathway can be blocked, and through signal crosstalk with non-canonical pathways that are also activated by mutation in HPV-positive cancers, result in stimulation leading to EMT and cancer stem cell maintenance. So what's important here is that the canonical response within the tumor is disrupted, but TGF-beta is secreted by the tumor into the tumor microenvironment where it has myriad immunosuppressive factors, including increasing CAS, increasing deposition of extracellular matrix, increased Treg, decreasing CD8 proliferation, decreasing APC function, decreasing M2 TAMs, and increasing angiogenesis. So it's reasonable, based on about 20 years of data of disruption of the TGF-beta pathway, that a TGF-beta targeted agent may work and augment the effects of PD-1, PD-L1 targeting. I'm struck by the duration of response and the effect on 1-year OS. I think it's extremely promising. You do get some increased toxicity from the combination. What are the next steps? Well, increasing the sample size in the ongoing phase 2 may tighten those confidence intervals, but we've been surprised recently in head and neck in confirmatory signals where we thought we were augmenting the effects of PD-1, PD-L1, that they weren't confirmed in a randomized phase 2, so I strongly encourage that that be done. We're looking forward, potentially, if the signal is confirmed, to combinations with chemotherapy, given synergistic data in vitro model systems of synergy, and in particular because a lot of the long-term toxicities are due in these diseases due to the long-term consequences of chemoradiation and induction of fibrosis, which is largely TGF-beta driven. The addition of these agents to chemoradiation platforms may not only potentially improve disease outcomes, but decrease toxicities as well. Thank you.
Thank you very much for that great discussion. I think we have a couple of moments for any questions. Well, Maybe I'll start with a question. So this was an excellent discussion of the potential role of TGF-beta in pathogenesis of these cancers. But I was also struck by Dr. Sullivan's comments about the unmet need for the majority of patients where these drugs don't work when we look at immune checkpoint inhibitors alone. Is there any role for TGF-beta and this kind of a drug in overcoming resistance in patients who have already progressed on PD-1 therapy?
That's a good question. So as part of the ongoing phase 2 trial, we actually have separate cohorts for patients with checkpoint naive and checkpoint refractory disease. So we're actually continuing to look at response rates or clinical activity in patients with prior checkpoint therapy, and that data is not mature yet. So it's something we're actively looking at.
All right, thank you very much. And I think we're going to move on to our final presentation and discussant. This is a clinical trial coming from Dr. Hyun Chung from South Korea, who is presenting pembrolizumab after 2 or more prior lines of therapy in advanced small cell lung cancer, which are results from Keynote 28 as well as Keynote 158. Welcome.
Shall I do it? Just takes a moment.
Oh, okay. Yeah. Thank you, Chairman. Good afternoon. On behalf of my co-investigators, it is my great honor to discuss with you about the combined data of pembrolizumab for advanced small cell lung cancer patient from Keynote 28 and Keynote 158 in this plenary session, specially dedicated to the memory of Professor Wang Gi-hong, who was a former alumni of Yonsei University, Seoul, Korea. These are my disclosures. Despite available first and second line therapies, 5-year survival rate for patients with extensive stage small cell lung cancer is quite low, less than 10%. There are significant unmet needs for patients who experience disease progression after first and second line treatment. Until approval of nivolumab in last August, there was no approved or recommended treatment for the third-line treatment. In a real-world analysis of the patient who received the various third-line treatment, the overall response rate was 21.3%, with the median duration of response of 2.6 months, and the median overall survival was 4.4 months, with only 11% survived after 1 year. The anti-PD-1 monoclonal antibody pembrolizumab has demonstrated anti-tumor activity as monotherapy in patients with previously treated extensive stage small cell lung cancer in 2 multi-cohort studies. Keynote 28 is a phase 1 study with study design of target enrichment that involves only PD-1 positive patient. Keynote 158 is a phase 2 study with all-comers disease, all-comers criteria that involve the patient regardless of PD-L1 expression level. The overall response rate of Keynote 28 was 33.3%, and it was 18.7% in Keynote 158. The duration of response in Keynote 28 was 19.4 months, which was not reached yet in Keynote 158. The PFS was 1.9 and 2 months respectively, and OS was 9.7 and 8.7 respectively. These are study design of Keynote 28. Patient was eligible if the patient has unresectable or metastatic small cell lung cancer who had progressed after standard treatment or they were ineligible for the standard treatment. The patient must be immunotherapy naive with good performance status, having more than one measurable lesion and no previous autoimmune disease or non-infectious pneumonitis. Patients showing higher or greater than 1% PD-L1 positivity were enrolled. Patient received pembrolizumab 10 mg per kilogram Q2 weeks until 2 years or until progression or until toxicity occurs. The primary endpoint was overall response rate by investigator analysis, and secondary endpoints are PFS, OS, duration of response, and safety. Exploratory endpoints was efficacy in biomarker subgroups, and response was assessed every 8 weeks for 6 months and every 12 weeks thereafter. In Keynote 158, this is the multi-cohort phase 2 basket study with 10 cancer types where small cell lung cancer was treated with pembrolizumab. The patient was enrolled regardless of PD-L1 expression level if the patient can provide tissue samples for the future biomarker assessment. The eligibility criteria was quite the same as Keynote 28. Patient received pembrolizumab in a flat dose of 200 mg every 3 weeks. The treatment schedule and primary endpoints were quite the same as Keynote 28. Response assessment was done every 9 weeks for the first year, and then every 12 weeks. Current analysis, the exploratory pooled analysis of efficacy and safety includes subset of patients from the Keynote 28 and the Keynote 158 who had received 2 or more previous lines of treatment. Responses were assessed for RECIST 1.1 by independent central review. Efficacy analysis includes overall response rate, duration of response, PFS, and OS. Response rate was calculated using Klopper-Pearson's exact binomial method, and survival was calculated by Kaplan-Meier method. Total 131 patients, 24 from Keynote 28 and 107 from Keynote 158, small cell lung cancer patients were treated with pembrolizumab. Among them, 83 patients, 19 from Keynote 28 and 64 from Keynote 158, received 2 or more prior lines of treatment. So we selected these patients for this combined analysis. As of July 2018, the follow-up duration was 7.7 months. 8 patients completed the study, and 2 patients are ongoing, and 73 patients stopped the treatment mainly due to cancer progression. In patient population, they are generally represent the advanced small cell cancer patient population. Median age was 62 with male preponderance, and 16 patients has stable brain metastasis. 64 patients has 2 lines of previous treatment, and 36 patients has 3 or more previous lines of treatment. PD-L1 positivity was 57%, and 80% received prior radiotherapy. The confirmed response rate was 19.3%, including 2 CR and 14 PR patients. Disease control rate was 37 patients, and among 16 responders, 14 patients showed PD-L1 positivity. Importantly, the response observed was durable. Among 16 patients of responders, 9 patients continued duration of response longer than 18 months. And the median duration of response not reached yet, ranging from 4 months to 30.58 months. As we see here, the most responders' response occurs within 2 to 3 months of the treatment initiation. And among 16 patients, 6 patients with green circle progressed, and 10 patients are still remaining in response. Among 10 patients, 9 patients stopped the treatment after 2 years of treatment, and 1 patient is ongoing in PR status. In waterfall analysis, among 75 patients who had at least 1 post-baseline treatment access, 44 patients showed tumor reduction from the baseline in the sum of the target region, and 29% showed tumor shrinkage more than 30%. 24% of Keynote-28 and 31% of Keynote-158 showed tumor shrinkage more than 30%, and their shrinkage pattern was similar between the 2 cohorts of Keynote-28 and Keynote-158. With a median follow-up duration of 7.7 months, the median PF was 2 months, and 1 year and 2 year PFS rate was 16.9% and 13.1% respectively. The median overall survival was 7.7 months, and 1 year and 2 year overall survival rate was 34.3% and 20.7% respectively. When we turning our interest to the safety, 61% of patients experienced a treatment-related AE, including 9% of grade 3 to 5. 2 patients expired from pneumonitis and toxicolitis. 24% of patients experienced the immune-related AE, including 6% of grade 3 to 4 toxicity. When we compare the 2 groups about the toxicity pattern and the grade, there were no difference between the groups with patient 1 or more prior lines of treatment and between the patient among the patient with 2 or more lines of treatment. These are the treatment-related AEs. The most common treatment AE was fatigue, pruritus, rash, followed by hypothyroidism and arthralgia. 1 patient experienced grade 3 asthenia, and there was no difference between the 2 groups in the grade and frequency of the side effect. Most common immune-mediated AE were hypothyroidism, followed by hyperthyroidism and infusion reaction. Grade 3 immune-related AEs were colitis, adrenal insufficiency, pneumonitis, and pancreatitis. In this pooled analysis, pembrolizumab had anti-tumor activity among patients with extensive stage small cell lung cancer who had received 2 or more previous lines of treatment. Responses were durable, and most responses lasted longer than 18 months. Median overall survival and PFS in this pooled analysis were similar to the small cell lung cancer cohort populations of Keynote-28 and 158. No new safety signals were identified. This pooled analysis supports the use of pembrolizumab monotherapy for patients with extensive stage small cell lung cancer as 3rd line or later treatment. On behalf of study coordinator, co-investigators, and study sponsor, I'd like to thank to the patient and their family and personnel who actively supported this trial. This study would not be possible without their strong support. Thank you for your attention.
Thank you very much, Dr. Chung. And last but definitely not least, I'd like to welcome my friend Teofik Wanekoko, who is a drug development expert in experimental therapeutics and lung cancer, to give his perspective on this exciting work.
Thank you to the program organizer and to the chair as well. It's my honor as a second-generation mentee to Dr. Waun Ki-hong to be able to discuss this important abstract presented by Dr. Chung. So to start with, I think in order to appreciate the importance of the result just presented, it would be good to understand where we stand when it comes to 3rd-line treatment of patients with small cell lung cancer. And when we're looking for prospective validated data, the honest truth is we actually do not have much. mostly because nobody has been bold enough to want to conduct definitive randomized trials in this population of patients. And so we've relied mostly on institutional data, observational data, to sort of get a glimpse of what is going on with these patients. So prospective observational data from Germany in 432 patients highlight one important fact about these patients. So 432 patients receiving frontline therapy were then followed to see how many of them will make it to third line. As you see here, only about 20% of those patients actually made it to third line treatment. Recently, we looked at our own institutional data here at Emory, here in Atlanta. In 528 patients coming to our institution and looking at how many of them, number one, got treated, and how many of those patients made it to third line. What is important to realize about this, as we all recognize, is half of these patients actually did not get any systemic therapy, which is one of the main challenges that we face when it comes to treating patients with advanced stage small cell lung cancer. But if a patient is able to make it to third line, we see that these are actually the patients that do the best. Now, is that because they got third line because they survived, or is it because the third line treatment is making them live longer? Until we have prospective data in this population of patients, this is a question that we'll continue to struggle with. Only 6% of our institutional patient population, however, made it to third line therapy and beyond, suggesting to us that this is an important population of patients, but the impact of what we are doing in this population of patients will probably be stronger if we can move it into the earlier stage setting. So what is the key lesson that we can derive from the study just presented? Is that pembrolizumab as a single agent does have activity, mostly in terms of impact in overall survival. If you only go by progression-free survival data, it looks modest, but whatever this is doing is translating into overall survival advantage for patients. And therefore, in heavily pretreated patients, as we've already known with nivolumab, which was also tested in the same setting, this provides additional confirmatory evidence, although not randomized study, single-arm data that is tracking what we already know with other immunotherapy agents in this population of patients. It's also important to realize that the safety was shown in these third-line patients to be comparable to those patients getting this as second-line therapy. One of the reasons why patients do not survive long with small cell is they are not able to tolerate treatment in terms of toxicity, but also because at the time of progression, majority of these patients are too deconditioned to be able to proceed with further treatment. So it is reassuring that giving this agent in the third line or beyond did not confer additional toxicity on patients. When we look at the data from the third-line nivolumab trial, this curve actually looks similar to what Dr. Chung just presented. Progression-free survival impact is modest, but overall survival impact is much more significant, leading to the approval of nivolumab as a third-line therapy for this population of patients. And I think the data that we just saw now further confirmed the initial evidence that we got from nivolumab, although both of them were uncontrolled studies, but now that we have 2 or more studies showing the same trend, I think it becomes more reliable that immunotherapy in this setting will be beneficial to patients. Only a small subset of these patients do achieve objective response, but when they do, it is very, very remarkably long. And it's comparable to what we saw with nivolumab. If you're able to induce response in this patient, they're likely to maintain this response for a long period of time. What is relevant, though, is at what point do we want to use this agent and in what subset of patients? Can we identify who these responders are? So this is a summary table, which is not to compare one study to the other, but just to give us a trend of what I view as the impact of immunotherapy in the second line. So when we look at the original Keynote 028 and 158, including all those with second line treatment as well as third line, and the recently presented study by the French group of atezolizumab strictly in the second line, we see that the survival impact of immunotherapy is stronger the earlier you use it, even though you still see activity in the third line, but the overall survival benefit is stronger in patient getting it much earlier in line. So quick points to ponder in terms of the data that we just saw. We have to be aware of immortal bias when we are dealing with long-term survivors, meaning patients that will do well regardless of what we do to them. Is this what we are dealing with when we're dealing with patients receiving third-line immunotherapy? And can we identify the unique biology of this subset of patients that are responding and living long and able to tolerate third-line treatment and beyond? And more importantly, how relevant is this data to our current day clinical practice as well as ongoing research in a very rapidly changing treatment landscape for small cell lung cancer patients? The search for predictive biomarker in small cell, as we've heard for non-small cell earlier today, continues. PD-L1 is actually less relevant in small cell compared to non-small cell, in part because very, very small proportion of small cell tumors will have PD-L1 expression, less than 10% of them, depending on how you set your cut point. And even when you look at not just tumor, but stromal expression of PD-L1 with tumor-infiltrating cells, that is only in about 40% of patients, going by the experience from the Keynote 158 study. Efforts to use tumor mutational burden in the Checkmate 032 study showed some promise, but this was a retrospective analysis showing that high tumor mutational burden might predict for better efficacy of immune checkpoint blockade. This requires prospective validation, and hopefully we'll have this data in the coming years. A genetically engineered mirroring model of small cell also showed that this is an inherently cold tumor. And what you see in the animal model of this disease is you do not have direct tumor infiltration by lymphocytes or immune cells. Oftentimes what you see in some is just tumor-stroma interface infiltration with restricted presence of immune infiltrate within the tumor microenvironment itself. Recently published data from the parent Keynote 028 study also showed that the gene expression profile looking at T-cell inflamed signature may help identify who responders are to pembrolizumab, and also this tracked very well with the impact on progression-free survival. Similar data had been shown before by Dr. Gajil when they look at pembrolizumab in the maintenance setting, and they showed that the presence of tumor immune infiltrate in the tumor-stroma interface correlates well with progression-free survival and resulted in overall survival advantage in this small subset of patients, suggesting that this could be a potential biomarker that will help us identify patients going forward. So in the last few minutes that I have, I want to talk about the implications of this data we just saw for clinical care and research. The first one is the landscape of small cell lung cancer has changed in the last 1 year, where we now have the use of immune checkpoint blockade along with chemotherapy as part of our frontline regimen, which then calls to question as to what do we do for patients after they've progressed on a regimen that contains immune checkpoint inhibitor. From the Empower 133 study, we know that the addition of atezolizumab to chemotherapy led to better survival outcome. But we do not have any reliable biomarker, be it clinical, demographic, or biochemical, or genomic, that we can use to identify those patients that will benefit from immune checkpoint blockade. So we are stuck with treating all these patients with immunotherapy. If they progress, what then do we do? And I think that is the most important question at this point. What is the optimal salvage therapy for our patients after they failed immunotherapy and chemotherapy? Now, Empower 133 is not the only study. We have 2 other large studies that will read out in the coming year, hopefully. And if they are positive, then that would then tell us that virtually all the currently available immunotherapy agents will be applicable in small cell lung cancer for frontline therapy. The French study looked at patients progressing on frontline platinum doublet chemotherapy and compared the use of chemotherapy in those patients who had very good response to atezolizumab. And this study actually showed us the way to go and what not to do. When you look at patients with platinum-sensitive disease, randomized to get chemotherapy or immunotherapy, in this case atezolizumab, those patients treated with chemotherapy actually did better in terms of progression-free survival. While the overall survival advantage of immunotherapy appear to catch up later on, I think the approach would be for this patient to consider them for retreatment with platinum doublet, but whether you then do that along with immune checkpoint blockade or not remains to be seen. The way I think about this going forward is if we're going to use immune therapy in the front line for all of our patients, those with durable CR or PR will most likely get retreated with platinum-based doublet, and whether or not we add immune checkpoint blockade at that point is probably debatable. But when they progress in the third line setting, I think the current study will give us a platform on which to build by looking for additional agent that we can then combine with immune checkpoint blockade. For those who did not respond to frontline therapy, of course, platinum agent will no longer be an option for that. And that would be where we might be able to use non-platinum cytotoxic agent. And since those patients will be coming off of immune checkpoint blockade anyway, I do not see any value at this point in using immune checkpoint blockade at that point. But in the third line again, after you've reset the thermostat for those patients, it will not be unreasonable to explore the use of immune checkpoint blockade plus a novel agent. While it is disappointing, I think we do have things to work on. We now know that high TMB or gene expression profile could help us identify those that we can target with immune-based strategies. MYC amplification could be predictor of aurora kinase inhibition, as we previously presented. And SLFN11 is a promising biomarker for DNA damage repair pathway that have been previously shown in small cell to be relevant. DLL3 targeted agents are in various phases of testing at this point in phase 1 and 2, although early definitive studies do not look as promising for ROVA-T. And we still have some kinase inhibitor strategies based on activating mutations that are not very common, but we can still see them in small cell. So in summary, I would say this study reproduced the efficacy of checkpoint blockade for relapsed small cell in the third line and beyond. It's showing very prolonged durability of response in those responders if you can find them. And it offers an alternative strategy beyond cytotoxic chemotherapy going forward. There are limitations to what we know. A single arm study without control, we have to be wary of immortal bias. Only a small subset of our patients currently make it to third line, but hopefully we'll be able to get greater number to that line. There is no established predictors of benefit, but there are promising angles that we can pursue. And despite the change in treatment landscape, I still think there will be opportunities for immune checkpoint blockade post-frontline. And with that, I thank you for listening, and I'll be happy to take questions.
Thank you very much. Are there any questions from the audience? All right, I will ask one question before we close the session. So, oh, never mind. I will. Please go ahead.
Gandhara, Davis, California. Can you hear me?
No.
Hello? My question is actually to both of you, and that is in both this study and the small cell literature with checkpoint immunotherapy, are the long-term survivors patients who responded? It may seem like a simplistic question, but in non-small cell lung cancer, 40% of the 2-year survivors, their best response was stable disease, and many of them, their best response was progressive disease. So this means there is a biologic discordance, at least in non-small cell lung cancer, between response rate, progression-free survival, and subsequent survival. So in small cell, is that what we're seeing as well, as you don't have to have a RECIST response to be a long-term survivor?
Okay, thank you for the question. Actually, there is some biological difference between the non-small cell lung cancer and small cell lung cancer, we think. So for one example, the PD-L1 staining. If we stain the PD-L1 positivity, even if we do the same method, small cell lung cancer shows only 30% of PD-L1 positivity, and non-small cell lung cancer shows 51% of positivity. And the main difference came from the immune cells. The frequency of PD-L1 positive is quite low in small cell lung cancer compared to non-small cell lung cancer, and the grade is also low. Most patients in small cell lung cancer, the CPS score is between 1 to 10. Very rare patient shows higher than 50. So there is some difference with more immune cells than less tumor cells, and this kind of harsh microenvironment can change the tumor response. Maybe that's the reason why there is less effect of stable disease in small cell lung cancer patient. Only responders shows long-term survival. Most of the stable disease, they make progression in a certain time of period.
Thank you, Dr. Gandhara. I think going back to your specific question about the discordance, if you look at everything that we've seen, there is definitely a strong discordance between the PFS and the OS. And I don't think we've looked at the data carefully enough to be able to translate that into the response. But one can at least make the assumption that if you do not respond at all, knowing what we know about small cell, things are not likely to work out well. But there is, for sure, a strong discordance between what we see in terms of overall survival and progression-free survival, suggesting that it's not just those who respond that is benefiting.
All right, and with that, I'm going to close this fantastic session. I want to thank all 4 of our presenters and all 4 of our discussants, and I wish you all a wonderful meeting. Bye-bye.